I think we are good to go. I got the thumbs up. Hi, everyone. Welcome. My name is Tim Griffin. Have the pleasure of hosting our speaker and introducing our speaker this week. So I'm going to give a brief introduction and bio, and then I'm going to hand it over to him. So our speaker is Malachi Griffith from Washington University School of Medicine. So Malachi is Canadian by birth. Got his Bachelor of Science from the University of Winnipeg in biochemistry and biology. So he's got some sort of Minnesotan, I guess, connection just north of the border from us. So, and from there, he got his PhD in medical genetics and bioinformatics at the University of British Columbia in the distinguished laboratory of Marco Mara. He did a brief postdoc then at the BC Cancer Agency for Genome Sciences. Center, and then from there went to Washington University, where he is now a professor of medicine and genetics. He's been very well funded, and as you're going to see, has some really cool and amazing work going on. I got to know him and have gotten to know him. I think he's been one of the core members of the informatics technology for cancer research, the NCI program. Has been funded very well through that, as well as other things. He received a V Scholar Award as well. So we've been doing some work with that ITCR program as well, and have gotten to know what he's doing, and it overlaps some of the interests that we have as well. So we're really excited to have him here, and I will give the floor to him.
All right, thank you. Thanks for the Cancer Center hosting, and thanks so much to Tim for thinking of me for this invitation. I'm really honored to come here and chat with you. This presentation is really focused on a major area of interest in the lab. Quite a lot of this presentation is being presented for the first time. So if you have any questions or some things that are unclear or feedback, I welcome your questions at the end. My lab is really computational biology, bioinformatics, genomics focused. So all of this is kind of from that perspective of sort of trying to apply bioinformatics methods to a translational medicine research problem. So I titled my talk, Basic and Translational Applications of Neoantigen Discovery Resources. But I will, I guess, start by saying that this presentation is going to be quite focused on the translational end of things with a little bit of background. These are my potential conflicts of interest that are related to this topic. Long story short, it boils down to consulting activities for various commercial entities that are also working in the neoantigen therapeutic space. Okay, so this is going to serve as a roadmap. So I'm going to keep referring back to this really high level kind of cartoon depiction, which is going to be essentially the overview for this whole talk. So I'm going to start with some basic background, some of which is probably quite familiar to some of you, maybe less to others, depending on what area of cancer research or practice that you're focused on. I'll very briefly cover sort of some example preclinical study work, and then I'll really get into kind of the main thrust of the presentation, which is to sort of walk through this process of trying to design and deliver a neoantigen targeting therapy that at a high level involves taking a sample of a tumor, characterizing it by deep sequencing, identifying the molecular features that are unique to each patient's tumor, predicting which of those features might be useful to elicit an immune response, selecting the best candidates based on our current knowledge, and then manufacturing some kind of T-cell related therapy, of which there are several options, but the one that we're really going to focus on today is personalized neoantigen vaccines. Depicted here, it shows mRNA vaccines. That's sort of one of the ways, perhaps one of the more famous ways of delivering this kind of vaccine. And it's one of the platforms that we're using a little bit, but it's just sort of one example. And we also use peptide and DNA delivery approaches as well. I'm not going to talk about some of the other types of therapies that could involve neoantigens, but a lot of the workflow is sort of equally applicable to those. So you could imagine picking neoantigens, targeting them with the vaccine, but you could kind of take the same output and think about how you would create a personalized T-cell therapy with that same output, or design other sort of more difficult to design targeted therapies like bispecific antibodies or TCR mimics and so forth. But assuming you can figure out that manufacturing part, you'll then hopefully deliver that therapy to the patient, hopefully in a way that invokes their immune system to attack their tumor. We're doing this in the context primarily of clinical trials. And so I'm going to talk about some of what it's like to facilitate those clinical trials from a genomics and bioinformatics perspective, and then how we think about validating the results and performing immune monitoring to get a sense of, is it working in the relatively early stages of these clinical trials. Okay, so I'm going to do a little bit of a high-level background, but really not too much. Starting with this basic premise that T cells can and do kill tumors. And we know this pretty convincingly from some success stories in T cell therapy, to sort of really prominent examples of which are CAR T cell therapy and checkpoint blockade therapy. So both of these therapies involve, well, in the case of CAR T cell therapy, involves bioengineering a T cell to recognize something that's expressed on the surface of a particular type of tumor cell, using the patient's own T cells to create this bioengineered therapy, expanding those cells, infusing them back into a patient where further expansion occurs, usually as a single bolus treatment where you have a kind of nuclear bomb of T cells. So huge numbers of these things that have been, in some cancer types and some patients, very successful at killing tumors. And then a related therapy, checkpoint blockade, which has also been very prominent in the last 10 years, essentially works by overcoming the tumor's natural resistance to the immune system by blocking the signaling molecules that essentially tell the immune system that a tumor cell is not foreign, that it shouldn't be attacked. So you're kind of like removing that inhibition so that the immune system is able to recognize and attack tumors again. And this has also been very successful in some types of therapies, and pretty extensive research at this point has identified that it's the neoantigens, the tumor-specific antigens within a tumor cell that allow these therapies to work. We're not targeting them directly or in a kind of rational way, but it's really neoantigens, tumor-specific antigens that underlie the response to checkpoint therapy. So I'm going to talk about neoantigen vaccines primarily, which is kind of an alternative or complementary strategy to these two T cell therapies. Okay, so what is a neoantigen? If you're not familiar, so a neoantigen in its simplest form is just a single amino acid difference acquired in the DNA of the tumor that creates this amino acid change that can be recognized as foreign by the immune system. So in this very simple example, we have a single amino acid substitution caused most likely by a single DNA nucleotide substitution that results in a potential difference between normal cells that do not have that somatic alteration and tumor cells that do have that somatic alteration, allowing the immune system to potentially detect the tumor cell as foreign and something that should be attacked, much like the immune system would attack a cell that was infected with a pathogen. These neoantigens, I showed an example of the simplest form, but they come in many, many possible classes. So point mutation is sort of that example I showed on the last slide, but it can be something that's a little bit more dramatic, like a deletion or an insertion. In some cases, those deletions or insertions can lead to a frame shift and potentially like a good stretch of amino acid sequence that's aberrant and only in the tumor cells, not in the normal cells. Alternative RNA splicing can similarly lead to either in-frame or frame shifting transcripts that give these tumor-specific sequences. Intron retention can similarly give rise to large chunks of frame-shifted sequence. Gene fusions that happen when chromosomes are rearranged and sort of fuse two genes together can give rise to either in-frame or frame-shifted fusion partners. And then there's some more kind of exotic forms that involve aberrant reactivation of the expression of something that's not normally expressed beyond, say, the earliest stages of development, or whose expression is really normally limited to a very specific cell type. Other sort of non-canonical sources are just expression and translation of regions of the genome that just really normally aren't expressed in healthy cells at all. Post-translational modifications where the amino acid sequence is the same, but there are additional modifications on those amino acids that create enough difference that they could be recognized as foreign by the immune system. And the way all of this works in terms of the cellular machinery is that you start with one of these tumor antigens that arose from one of those many possible sources that I depicted on the last slide. So it's inside the cell of the tumor cells. It gets processed by the proteasome into smaller antigenic peptides, transported into the endoplasmic reticulum, where it is assembled, if all of the pieces kind of fall into place, onto an MHC molecule. And the MHC molecule is trafficked to the surface of the cell, where it has the potential to be recognized by a T cell receptor on a CD8 positive T cell. And if this happens, it can trigger a cell killing response and expansion of T cells that have this T cell receptor that recognizes this peptide MHC. So this process is happening all of the time in all of our cells, and it's one of the things that allows the adaptive immune system to work, so that if something winds up in a cell that's not supposed to be there, the immune system can recognize that foreign antigen and kill infected cells. So we're really just kind of taking advantage of what happens already in cells that are infected by a pathogen and sort of repurposing that idea for tumor cell therapy. Here's a 3D depiction of the major components of that MHC complex that's depicted on the bottom right here from the previous diagram. It's really made up of three main parts. There's the MHC molecule, which is interesting from a genomics and genetics perspective, because the HLA loci that encode the MHC molecules are among the most genetically diverse in the human population. So there are many, many different versions of the HLA genes. Everyone in this room probably has a unique set of just the class 1 HLA A, B, and C genes. Each of us has one copy from our mother, one copy from our father. You may have heard of HLA typing in the context of organ transplants. This is another context we think about this. This is one of the ways that we determine whether someone is a suitable donor for tissue from another person. We try to match them based on how similar their HLA alleles are. At present, there's over 5,000 known alleles, but there's probably 300 or 400 that occur at reasonable sort of clinically relevant frequencies in the human population, but that's already a huge amount of diversity. And each of those MHC molecules has a different behavior because of those sequence differences. And the thing that they do is bind peptides so that they can be presented on the surface to the immune system. The class 1, depicted on the left here, the class 1 MHC complex holds peptides that are around 9, but they can be sort of 8 to 11 amino acids long usually. And it's generally thought as a rule of thumb that each MHC molecule will kind of fit about 1% of the peptides of that length that it's exposed to. So there's a real kind of lock and key sort of like mechanism happening here, where this peptide really has to fit 3-dimensionally into the groove of the MHC molecule. Formally, there's a huge number of possible peptides, 2.2 times 10 to the 14 possibilities. Of course, in the human proteome, that's quite constrained because the sort of raw starting material is what's actually in our genome and a little bit more what's actually expressed and translated. But when you start looking at those non-canonical things, there's a huge diversity of potential peptides that we could be looking at. And then the third major component is the T cell receptor. For CD8 T cells, the T cell receptor, along with the CD8 complex, forms this whole complex of recognition. And the business end of the T cell receptor is created by this really amazing genomic recombination mechanism that sort of scrambles up the sequence of the recognition part of the T cell receptor to give an almost infinite diversity of T cell receptor sequences at that end that's going to try to recognize the peptide MHC molecule. And this is what allows the adaptive immune system to really work, to recognize foreign pathogens that can't be predicted in advance. You have to have this ability to create this sort of random diversity of sequences and structures so that the adaptive immune system can work. Okay, so that's the background. That's sort of the crash course and sort of the basic immunology of what we're thinking about here. Now I want to briefly pivot to preclinical study work. So to really work this all out, sort of a ton of really important mouse studies primarily, but also in other species have been done. There are some clinical trials involving companion dogs, for example. They're trying to mimic the same idea, though. It's sort of, can we take a mouse tumor? Can we identify the unique tumor-specific antigens in that tumor and then design a therapy and then treat mice that have those tumors or that get those tumors. And then try to understand why our targeting worked or didn't work. So as part of our work in the lab, we've really tried to support this preclinical work. We've created a computational workflow for non-human species. As I said, it's mostly optimized for mouse models that takes sort of raw sequence data, performs alignment QC, somatic alteration detection, and then feeds those alterations into a workflow that tries to prioritize antigen targets or candidates that could be used to try to treat the mice with either an engineered T-cell therapy or a personalized vaccine that's sort of unique to the characteristics of that mouse's tumor. So far, we've done this for 21 murine tumor models spread across 5 cancer types in collaboration with 7 different labs that we're working with, working in sort of different areas of tumor biology that they're focused on. Ranging from sort of pancreas, lymphoma, sarcoma, lung, and breast, which largely mirrors sort of what human clinical trials we're working on. And then just to give you kind of one example of what this looks like, this is sort of a one-slide depiction of the whole experiment. So in this example, we're starting with the KP2 model, which is a pancreas cancer model. That's created with a series of targeted genetic engineering. These cells will form tumors essentially just because of those specific driver alterations that they've been given. But then they really don't have that many other mutations. They form tumors relatively quickly and they don't acquire a ton of other somatic alterations. So to make them a more useful model for human tumors that tend to have more mutations, pancreatic cancers that have more mutations, they've been exposed to DNA damage to create sort of subclones that have the original genetic engineering, but then have acquired additional mutations. And those are used in mice. So in this, there's a lot of different experimental approaches, but in this approach, the mice are given a vaccine before they get the tumor, then the tumor is implanted, and then they get booster doses of the vaccine, and there's been various schedules experimented with, and then the tumors either grow or they don't grow a certain amount, and then you take down the mice and assess tumor burden, and then do flow cytometry and ELISpot to try to get a sense of, is the immune system actually recognizing and potentially killing the tumor. In this example, which is really led by Rohina Pani, a surgeon at WashU, she chose from the output of our workflow 7 neoantigen targets that she thought looked like the best candidates, encoded those into an mRNA vaccine and used that as the vaccine to treat the mice. So with a series of mice, she first just did basic tests of tumor implantation followed by vaccination to show that with vaccine alone, the tumors do seem to be controlled to some degree. So they're growing more slowly when they're treated with the vaccine compared to the vehicle. So the immune system has been primed to recognize this tumor is killing the tumor, and then it gets boosted, kills the tumor even more. This results in a statistically significant reduction in tumor weight. And then if you take those tumors out, you can characterize them, and you can also characterize T cells from the spleen or the periphery or from lymph nodes to try to get a sense of whether you can detect neoantigen-specific T cell responses. And this is done with an ELISpot assay that I'll sort of go into a bit more detail later. And then each individual component, the 7 neoantigen targets that made up the vaccine, is tested individually to try to get a sense, was it one of those that seemed to be driving response? Was it all 7? Was it none of them? In this case, it really seemed to be 2, kind of 3, that are eliciting immune responses, and the others really don't seem to. And so this creates an interesting opportunity to start to ask what's different about the ones that give a response versus those that don't. So from the bioinformatics tool development perspective, we're really keen to go and look at these and try to figure out what's the pattern, because we thought they were all good candidates. They were all ranked very highly and met all of our criteria, but less than half of them actually induced an immune response. And why is that? Okay, so this preclinical model is being done sort of over and over again with all kinds of different variations and delivery approach and composition of the vaccine and timing of the vaccine and combination with adjuvants or immune stimulating molecules. A general theme of these studies is that you will see this sort of partial control of tumor growth with vaccination alone, but if you combine it with checkpoint therapy, you can achieve tumor elimination. Okay, so that's the preclinical sort of teaser, and then I'm going to pivot and focus really on the human application now. So as you can imagine, thinking about these kinds of improvements that we're really learning about in the preclinical models and applying those to human therapies is quite challenging. It's a complicated enterprise, and my lab is really focused on enabling the sort of bioinformatics and genomics side of that. So the overall workflow looks something like this on the left. So you isolate tumor tissue and matched normal tissue, usually blood, isolate DNA and RNA from those tissues, generate sequence data, analyze that data to identify the various kinds of tumor somatic alterations that might give rise to a neoantigen that's tumor specific. In parallel, figure out the HLA typing of the patient so you know which version of those MHC molecules each patient has, and then kind of put all the pieces together to try to predict from these alterations presented by the patient's MHC molecules what epitopes might be presentable that are unique to the tumor, and then use that information to design a personalized therapy and deliver the therapy to the patient. Right now, we have 12 clinical trials that are in various stages of, a lot of them are completed and have already, a few of them have already been published. Many of the others are still undergoing the sort of post-trial correlative analysis in preparation for publication, and some of them are underway and still enrolling patients right now. Overall, over the past several years, we've gone through this workflow over 200 times now to design personalized vaccines. About 2/3 of those have actually wound up being delivered to human patients spread across these trials. And of course, we're using the results of this to try to improve our algorithms that facilitate the predictions in the kind of the most sort of real applicable data that you can have, which is actual real humans with real immune systems responding or not responding to components of these therapies. So this is one example. I'm going to show two examples of what these trials can look like in terms of design. You start with, in this case, it's triple negative. So quite a few of the trials, as I showed in the last slide, are focused on triple negative cancer, which, as I'm sure many of you are aware, is a breast cancer that has very poor outcomes and not great effective therapy options right now. So we isolate samples from these patients, go through this personalized vaccine creation approach that I described. And then in this case, it was a DNA vaccine. So the epitopes are encoded into a DNA vector, which then undergoes manufacturing and safety testing and is delivered to the patient somewhat remarkably via an electroporation device, which is sort of zaps your arm kind of at the same time as giving you the vaccine. In this case, there were 7 doses of the vaccine, again, arranged in kind of like a prime boost sort of schedule. So they get 3 doses in relatively quick succession, and then the remaining doses are spread out a bit further. This trial was published at the end of 2024, showing that this approach was effective and safe in terms of eliciting immune response. It was a phase 1 trial, so there's not a ton we can say about efficacy of the vaccine, but it was able to induce immune responses, and I'll show some of those results a bit later. And then the second example is pancreatic cancer, which is probably the next most major cancer type that we're focusing on. In this case, this is a more recent trial. The trials are gradually getting more complicated. This one actually has 2 arms, experimenting with the idea of whether it makes a difference to give a neoantigen vaccine after surgery or before surgery, which means that these patients either get a chemotherapy holiday and then their vaccine, or they get their vaccine right away. And the idea is to, again, assess whether this is a safe approach and whether there's any difference between these 2 timings and just whether it's working overall in terms of immune response. This trial, in contrast to the previous one, is using peptides as the delivery approach instead of DNA. And I'll talk a little bit more about what the peptide delivery system is like. Okay, so that's the background for the trial. And now I'm going to kind of try and walk through using examples from those trials, what it's like to go from the raw data to actually designing and delivering these vaccines. So it starts with many things upstream of this, consenting the patients, obtaining samples and so forth. But the part that my lab is focused on the most really starts with the sequence data. So we have at WashU a CLIA-CAP lab that does IDT exome sequencing for this kind of study. We pair that with tumor RNA sequencing. We do this in an unusually sort of comprehensive way. So compared to sort of research RNA-seq, where you might get 300 or 30 or 40 million reads to get a decent estimate of gene expression, we really sample these transcriptomes very heavily. And similarly for the DNA, we sequence very deeply to make sure that we don't miss any variants in tumors that might be of variable purity, which is definitely the case in tumor types like pancreatic cancer. In parallel, we also perform clinical grade class 1 and class 2 HLA typing. And then we also analyze the sequence data to get the HLA typing, and then we compare those results both as a kind of like sanity check and QC measure, but also in some cases to identify discrepancies for some of the less well characterized HLA alleles. So this is kind of just to give you a little taste of what the data looks like. So this is an example from a recently completed pancreas trial where there were 29 cases that went into the sequencing. There was some attrition at the sequencing stage where there was just the TMB was too low, tumor mutation burden was too low in one case, and some cases just had too low tumor cell content to really detect variants. But most cases worked, and we sort of through our interactions with the FDA have gradually just defined sort of metrics for quality that ensure sort of robust sensitivity for each patient. So we're really shooting to make sure that we're exceeding our target sequencing metrics, which in this case, we're shooting for about minimum 250x coverage of each base in the exome, and we're getting over 600x coverage. And then, as I said, very comprehensive RNA sequencing. We actually also, in addition to sequencing the RNA very deeply, we perform a cDNA capture of the RNA library using the same exome reagent to further concentrate reads on the regions that we're the most interested in, which is the protein coding exons. Again, using this trial as an example, this gives you sort of a bird's eye view of what the HLA diversity looks like for one of these groups of patients. So just looking, for example, at the three class 1 HLAs, A, B, and C, we're seeing in a cohort of 20 patients, 13 to 19 distinct alleles for each of the three class 1 alleles. And then, of course, the combination of those three is totally unique to each patient. Similar degree of diversity for the class 2 HLA alleles, sort of further emphasizing the need to really personalize the predictions to each patient's HLA complement. Okay, so we have this raw data, huge amounts of sequence data, up to, it's approaching 1,150 base pair reads of sequence data generated for each patient. How do we process and analyze that raw sequence data to actually come up with the vaccine design? So over the years, we've created what we hope is becoming like a quite robust, reproducible, portable, and reusable workflow to support this whole enterprise. On the top here is just kind of another depiction of this workflow where we start with raw sequence data in FASTQ format. We set up an automated pipeline run that really tries to do a lot of what I described: HLA typing, epitope prediction, variant calling, all of these steps as quickly and in as automated fashion as possible, but not completely automated, as I will discuss. We recently made this available as a preprint, which we're submitting for publication, sort of an attempt to describe sort of the end-to-end workflow. If you were to try to set this up yourself, how would you produce the sequence data, analyze the sequence data, run our workflow, and go all the way to the end of designing a neoantigen therapy? For those of you that are interested in the computational details, we've implemented this as a workflow definition language pipeline, which is sort of a formal way of specifying pipelines, their inputs, outputs, and results that you want to keep. Everything runs in Docker containers so that we can isolate dependencies between different softwares and keep it running over time. We use Cromwell, which is a piece of software that was developed at the Broad that sort of executes the whole workflow, and it allows us to run the exact same workflow on different computational infrastructure. So for our trial cases, we'll use the Google Cloud because it's the most performant, and we know that there's pretty much never any outages. But it also runs on our local compute cluster at WashU, and we've gotten it running on a compute cluster at Johns Hopkins that uses a different type of cluster. So I think we're starting to demonstrate that it's quite portable. Portability still of the complex pipelines like this is still actually quite difficult to achieve. Just to give you an idea of how much computational resources it takes, a single run for a single patient winds up using about 650 computers, 1,500 CPUs, a ton of memory, a ton of local storage disk. That sort of a lot of that is temporary for while the job's running, and then the final outputs are still quite large, and we've got this down to running in for a low TMB tumor. It can run in 24 hours, and sometimes it takes as long as 2 days. So really, to support that, like a main component of that workflow is really the part that sort of takes the raw sequence information and tries to predict neoantigens. So PVAC tools is the suite of software that really focuses on that part of the problem. So it's a Python toolkit that takes sort of variant information as its input and tries to predict what amino acid changes would arise from those DNA alterations, and then which of those would be good targets for immune therapy. This is all released publicly for free and as open access software, which we think is part of the reason why it seems to have good uptake, with many people downloading and using it and hundreds of citations of the related publications. We've really focused on, it's under continuous development. We've really focused on improving things like turnaround time by optimizing the computationally expensive algorithms and using cloud computing. The early versions of these analyses really focused on MHC binding. So the idea that a peptide has to fit in the MHC molecule, we think that, and everyone I think agrees, that this is necessary but not sufficient to induce an immune response, and that there's other things about those peptides that are really important, such as how they're processed and get to the point of being bound to an MHC molecule, but also other features that predict whether they will actually be recognized by the immune system or not. We've continued to work on different sources, supporting different sources of neoantigens, such as fusion and splicing, which I'll talk a little bit more about on the next slide. And then developed some software to help with visualization and prioritization. And then some pieces of the software are really directed at supporting the different delivery mechanisms, because they have divergent needs when you get to that point of manufacturing. The most recent sort of major release of PVAC tools was to add this component of detecting RNA splicing derived neoantigens. So here's a kind of a visual depiction of how that works. So the software looks for mutations that might influence RNA splicing. So here's an example of a splice donor mutation in P53. It then looks at the RNA data for an alternative splicing event that would make sense for that DNA splice mutation. And then it looks at the alternative tumor-specific isoform to try to predict peptide sequences that would arise from the skipping of an exon or the extension of the boundary of an exon, either breaking or maintaining the frame and giving you tumor-specific peptide sequence. And then once you have those peptide sequences, they feed into the same sort of neoantigen prediction algorithms that are used by all of the sources of neoantigens. Okay, so once you've sort of done that raw processing, you have the problem that you do need to make some choices in terms of what you're actually going to select to be part of the design. And as you can imagine, this is complex and involves consideration of many features. So again, if you think about just kind of a cartoon depiction of this, you have some somatic alterations that are giving rise to amino acid changes. And we really want to understand which of those are processed in the cellular machinery, which of them, oops, these are actually backwards, which of them are sort of spliced into known or alternative transcripts, whether they're clonal or subclonal, whether the peptides that arise from them differ depending on which RNA transcript is being expressed, whether that transcript gets translated into actual protein, and then sort of the downstream processing and loading onto MHC molecules, and then presentation on the surface of the cell, and then whether it will be recognized by CD8 or CD4 positive T cells. So you get all of these different factors to consider, and for quite a while we were kind of like juggling spreadsheets and sort of looking at information in sort of different sort of raw forms to try to get a sense of data quality and things like clonality, allele expression, binding, presentation, immunogenicity. We have over 15 algorithms sort of asking different parts of these questions, or in some cases trying to like competing algorithms that try to get at the same thing, and we try to get a sense of how robust the predictions are by inter-algorithm agreement, whether the mutant and the wild type version are both bound and whether that matters, how unique the peptide is, whether there's both class 1 and class 2 recognition, and so on. Because of how complicated this was, we created a dedicated visual exploration tool called PVAC-View that gives you a list of sort of preliminarily ranked candidates with sort of the top peptide arising from each somatic event as a starting point, but then you can sort of click on that and drill down into much greater detail. So all of the different subpeptides that might be presented by class 1 or class 2, and all of the algorithmic predictions and how it intersects with transcript structure, all of that detail. And then an interface that allows you to kind of record comments and record a record of what you thought was a good candidate, or why something wouldn't be a good candidate and should be rejected. You can go and see a demo of this at pvacview.org with some example cell line data that we've shared to sort of show how this works. So, okay, what does it look like to actually apply this in a trial? So here's an example from one of our recent trials, again, using the pancreas trial as an example, where we start with hundreds up to 200 or 300 somatic variants. And we look at those to try to understand, well, which of them might actually give rise to an amino acid change, which is what's depicted in purple and green here. And then we further look at the RNA data to try to decide, okay, which of these are actually expressed as a further way of prioritizing them. If you do the sort of really deep comprehensive transcriptome profiling that we do, we generally find that for sort of 40 to 50 or sometimes even 60% of the candidates can be confirmed as expressed, which is what's shown in blue here. But you can see now we've kind of like dropped our candidates down to tens instead of hundreds of things to consider. And there's kind of a parallel version of this that's happening for fusion events and splicing events. Just showing the SNVs and indels here for simplicity. In some cancer types, we find some common drivers. This is a very common question when I talk about this subject, is are there any things that are just like you could make an off-the-shelf therapy out of? Pancreas is probably one of the best examples of this, because virtually every pancreas tumor has a KRAS G12 mutation, rarely one of the other KRAS hotspot positions. If it's missing, I almost wonder if there's a data quality issue because it's so consistently present in pancreatic cancer. So this is an example where you could contemplate some kind of off-the-shelf design, really focused on the G12 amino acid changing neoantigens and how they intersect and are bound and presented by the HLA alleles that are common in the population. And this is being done by some companies, and there are clinical trials that are really focusing on sort of a monovalent design that sort of focuses on this off-the-shelf approach. But mostly that's not what we're finding. Mostly we find a completely different set of variants in each patient. So pancreas cancer, this will really be the only one that's shared. In other types of cancers, we might run a whole trial and never see the same neoantigen target twice in 10 or 20 patients. So they're really, generally speaking, entirely personalized designs for each patient. So once you've got the subset that are expressed, that's where sort of PVAC-View really comes in, is sort of how do we decide of the ones that are expressed, sort of what other additional criteria would convince us to prioritize them for inclusion in the final design. And there again, we have an attrition that's sort of about the top half are selected. If it's a really heavily mutated tumor, then we might be able to be a little bit more particular about what gets included or not. But in this case, we're seeing sort of an end result of 10 to 20 candidates for each patient's tumor. And then we get to the manufacturing stage. So the turnaround time here for this process up to this point is something like 4 to 6 weeks. If things are going smoothly, it could be like 3 to 4 weeks. And then manufacturing is probably the next biggest step, either by encoding your candidates in a DNA vector or mRNA vector, or by designing synthetic long peptides and having those manufactured. And the goal is to get those things and deliver them to patients, often as a multi-site vaccine injection. So here's an example of how it sort of worked out for one of our recent trials. We had 23 vaccines ordered, 22 were released ultimately. These vaccines are formulated as 4 pools of up to 4 peptides. They're delivered in these 4 sites. And then each of these vaccines has to be subjected to, obviously, a pretty extensive drug product testing for safety before injecting something that was manufactured into an actual patient. Over the years, we found that cysteine-containing peptides can be unstable in storage, so we try to avoid peptides that involve cysteine when we're doing the peptide delivery approach, whereas with mRNA or DNA, you wouldn't have that consideration. Inevitably, throughout this process, some patients are lost for various reasons and don't make it through to treatment. But in this case, we had a pretty high success rate, so 82 of these up to 4 peptide pools were released, with only a few failing the safety and integrity testing. This is what it looks like sort of at the pool level for each patient, and taking into account whether manufacturing was successful. So this is what it looks like, the thing that we actually give the manufacturer at the top here. So we give them like a 50 amino acid sequence with our predicted epitope sort of highlighted in the middle with the mutation indicated for class 1, and then in bold is a predicted class 2 sequence. This is sent to the manufacturer, who then chooses the subset of that sequence to make the actual long peptide and to be manufactured. And they have criteria related to what tends to be manufacturable, stable, soluble in solution. So in this case, again, we have an attrition of maybe 30% of peptides are lost because we tried to synthesize them and weren't able to. And then the hash marks here show examples where the patient wasn't able to continue, or we had to drop one of the pools due to the safety testing step. Okay, so we're almost there. We've made it to the point of actually having that thing manufactured, shipped back, tested for safety prior to delivery, and then it goes to the patients. I don't have a visual depiction of that, sorry. But the next step is really to pretty much immediately start to think about, is it working? Can we detect an immune response? One of the common assays that's used to do this, and this is really, all of this work is led by Will Glanders in his lab, where we basically take the peptides that were given to the patient, we stimulate T cells with those peptides, and perform ex vivo ELISpots, and then also do further peptide by peptide stimulation to try to figure out which peptides induce an immune response, which ones don't, and whether the response is a CD4 or CD8 T cell response. This is what it looks like for a single patient. So we designed these overlapping peptides that contain the core binding sequence, in this case underlined it kind of in different registers to create some kind of internal controls. And then we look for evidence that there is more T cell immune response after vaccination than there is pre-vaccination, and compared to controls, and hopefully find that the peptide does indeed induce a T cell immune response. When you start to put this together for a whole patient, this is what it starts to look like with the pre and post vaccination time points here, just 2 time points showing this is kind of typical. So we're seeing sort of one peptide that seems to induce a very strong response, and then maybe 2 others that induce more modest responses, and then some that perhaps don't induce a strong response at all. And then this is done at many time points to try to gauge this sort of amplitude of response over time. So every time the patient receives a vaccination, blood is taken at that same time point, so that we can later do 6, 7, sometimes 8 time points to kind of see the response of the patient as they get the priming and boosting doses of the vaccine. So here's an example of one peptide that really seems to be inducing a strong response that's sort of growing over time as the patient receives additional vaccinations. And then putting all of this together and sort of summarizing it at the cohort level, we can see the sort of big picture is that in each of these 12 patients, in this case, the general trend is that we see immune responses growing after vaccination, usually multivalent, but not for every candidate that was in the vaccine, always for a subset. So 1, 2, 3, 4 candidates. I would say 4 out of like 20 is a pretty good success rate. 5 or 6 would be like really amazing. So there's clearly something that we still don't know about the prioritization, and this is what motivates us to continue working on the algorithms that nominate the candidates. So we have, we're being successful to some measure. We're getting in almost every patient, we get at least one candidate that induces an immune response. So this is a summary from another trial, in this case in triple negative breast cancer, where in 80% of the patients, we got 1 or more immune responses to individual components of the vaccine, but quite a few components that didn't induce immune responses at all. And so we're really thinking about what are the patient level and neoantigen specific features that are governing whether we will get a response or not, and then trying to bake those into an improved version of our prioritization schema and algorithms. At this point, many of you are probably wondering and should be wondering, this is all sounds nice, but does it actually work? So most of the clinical trials, I think I showed you 12 clinical trials on that overview slide. All of those investigator-initiated trials at WashU are phase 1 trials, and even doing that is incredibly expensive and complicated. We don't have the resources to run a phase 3 clinical trial. So a lot of the outcome of our trials is safety and feasibility, and we can measure immune responses, but we can't really do much in terms of survival analysis on these small cohorts of sort of 10 to 25 individuals. And also mostly these trials are single arm, or there's 2 arms, but it's not really like a control arm in the traditional clinical trial sense. But there are at least two phase 3 clinical trials currently underway. So we're eagerly anticipating Moderna and Merck's data, which should be becoming available before too long, within the next couple of years, I would say. I believe these trials are well underway and may have even completed enrollment, but of course it takes quite a while to complete the therapy and to monitor and for outcomes. Okay, so that's sort of the end-to-end process. We think that there's lots of room for improvement. Our ongoing developments are quite focused on immunogenicity as the sort of least well-represented data in our prediction algorithms, because it's the hardest to generate. So we think we're pretty good at predicting when a peptide sticks to an MHC molecule, and fairly good at predicting when the whole processing thing works and the MHC molecule makes it to the surface of an antigen-presenting cell or the tumor cell. But we're really not good at knowing which of those will be recognized by the immune system. So that's a major focus of the ongoing development. And then the technologies, underlying sequence technologies continue to evolve. So we're interested in integrating new data types. I didn't mention mass spec at all, which actually plays a really important role in all of this. So we're quite keen to integrate immunopeptidomics data, which is the data where you essentially like pull down MHC molecules, elute the small peptides off, and then use a mass spec instrument to try to identify what is there, sort of bypassing the sort of DNA, RNA focused prediction method that I was describing. There's a lot of interest in antigens that don't correspond like in this simple way to like a DNA change. There's all kinds of things potentially that are unique in tumor cells that aren't necessarily just arising from a single nucleotide point mutation. And then of course, we're continuing to work on just supporting these trials in the sort of most efficient way possible so that we can sort of continue to improve. These are the members of my lab that have contributed to this. Most of them, I kind of showed their pictures along the way. I didn't mention or show a picture of Obi, who's my twin brother, who co-directs the lab with me. And of course, is a major player in all of this. And none of this would be possible without our clinical collaborators who lead these clinical trials or the funding. I would love to take any questions.
I have microphones over here for anyone that has questions, please.
Questions here, and I guess we should look online if there's any in the chat, but I can start maybe here in the audience.
Yes.
So really interesting talk. I've got a couple of questions about how you've been able to perhaps look at efficacy here. And I realize some of the human clinical trials are ongoing, but maybe you have some information from animal models that might inform some of the stuff. So you mentioned that most of your vaccines are unique to each patient, but sometimes you had ones, the KRAS driver mutation, that showed up reproducibly. Is there any evidence that the ones with the driver mutations work any better than the ones that are just sporadic, just because every cell needs it, or is that just completely non-correlated?
Yeah, so I think it's actually a surprisingly controversial question. As you say, the drivers are really desirable from the perspective of you would not expect tumor cells to easily immuno-edit those things away because they are probably addicted to that sort of oncogenic effect. So that's sort of a plus. And it's definitely one of the prioritization considerations. So we're always looking like, are there drivers that we can include, even if, sporadic ones that are different in like a P53 mutation that's not the same mutation in every patient, but it might be a driver. Those are definitely favored in the design, but I don't know that we really know that they're better. I don't think we've seen a signal that they're more likely to be immunogenic that's really obvious yet, but we're maybe starting to get to the point where we would have enough data to start to get a hint of that. But it's also been suggested that the drivers, these hotspot drivers, are actually not good candidates because there's sort of like a tumor evolutionary expectation that they wouldn't be a driver if the immune system was good at finding them. And so you're kind of like stacking the deck against yourself. And there are kind of like people arguing both of these camps. And I think there's not really I think it's nuanced. I think for some drivers, it may be true that they're good immunogenic targets, but I also think that just bigger picture, there is something to that argument that if something was really strongly immunogenic, it wouldn't be a common driver in everyone's tumor.
So it's sort of a follow-up to that in terms of tumor escape from these things. So you certainly showed us some examples of efficacy in the early data, but it clearly looked like the tumors would still progress. Do you have any insights from any of these vaccine trials that you put forward to what's driving, what's the predominant mechanism of escape? Is it antigen loss? Is it tolerance induction? I'm guessing there's probably a component of both, but do you have any feel for how often one of these mechanisms or something I haven't even mentioned contributes?
I think the most sort of famously reported is presentation loss, but that is one of the easier things to get a readout of. So you can look for loss of heterozygosity at the HLA loci. You can look for loss of beta-2 microglobulin relatively easily. This is sort of like a straightforward way for the tumor to like stop all of this from happening. I think the question is the right question in a way. It's one of the biggest probably worries and caveats of this whole idea is that you have a method of therapy that relies on a pretty complicated cellular machinery with a lot of parts. And that creates a lot of like avenues for resistance. I think one of the reasons, or this is baked into some of the prioritization schemes. So for example, targeting candidates that are mutated and also amplified because it's difficult to like immuno-edit those away. Doing things that are like class 1 and class 2. So you have sort of like multiple of each of the alleles for both class 1 and class 2 to make it as kind of multivalent as possible. And then of course, like all cancer therapies, all of which do induce resistance, thinking about combination. So, and that is one area where this seems like encouraging. It seems that this is a therapy that would be added relatively easily to many existing treatment strategies.
Thanks.
But resistance is going to be a serious issue.
That was really great. I have many questions, but first, I noticed that in the phase 3 trial, both of them involved the use of PD-1 blockade as well as vaccine. And in your phase 1 trials at WashU, was that not included? And if you had included it, do you think more peptides would have scored as immunogenic, possibly?
Yes, I think the answer to your second question is I think yes, that is part of the reason why we don't see a response for a single peptide. It might be something about that peptide, but it might be a bigger picture thing about what's going on with the immune system. And checkpoint blockade seems like it creates an avenue to sort of expose more response capability. The early trials did have vaccine alone, but I think pretty much all of the trials that are underway now are being proposed involve combination with checkpoint. I think that's both the mouse studies and the human studies have shown that the combination definitely seems to be helpful. So I think that's, going to be common going forward.
So last thing, does your prioritization bake in the concept of having overlapping class 1 and class 2 candidates, or was that just what happened to be there that you showed us?
It doesn't strictly require it, but all of the delivery approaches we're using involve relatively long sequences with the hopes that we will have the capability to include class 2. And we always look at the sort of class 1 and class 2 predictions for each peptide and try to include in our long peptide parameters when we send them to manufacturer, hey, try to retain all of the sequence so that we have what we think is the best class 1 and what we think is the best class 2 sequence. I will say that the predictions for class 2 are, I think, much less developed. The tools have received a lot less focus. There's less training data. So I think those predictions are not as good. But it seems like from the readout from the trials that class 2 responses are important. So that's definitely a thought in the design.
I was just going to ask, following up on that, when you're assaying the peripheral blood over time, do you look at reactivity by CD4 and CD8 to sort of get at that question a little bit closer?
Yeah, I have a lot of worries about the assays that are used here. I think it's technically challenging to perform a class 1 or class 2 specific ELISpot assay. So there are different approaches there that folks are sort of experimenting with. There's a kind of blocking strategy where you try to use blocking antibodies to sort of enrich for class 1 or class 2 response. The strategy that's been the most used in our assays to date has been to do it class agnostic, and then to do a second step, which is to use some kind of cytometry to then assess, was it once we have a positive, did it look like it was a CD4 or CD8 positive? And then that's another area where the new assay development and sequence developments come into play, where in some cases we've gone and done single cell sequencing instead of ELISpot on these cell populations, and then you get to see cell by cell, am I a CD4 cell or a CD8 cell? And then the assay that we're kind of working to develop in collaboration with Johns Hopkins right now is the manifest assay, which is much more class 1 specific, where you're stimulating with just the core 9-mer class 1 peptide. And so you kind of get more of a clear readout, and kind of just all of those things together. But I think there is kind of some wishy-washiness in the immune validation response data that you're seeing in a lot of the publications right now as to whether it's really class 1 or class 2 or both.
And then you said the large phase 3 trials, you mentioned, are they incorporating the tools? You mentioned the immunogenicity being hard to predict, and I think obviously it would benefit from big data sets like that. I'm just kind of curious if that'll be open out there, or
I wish I knew. These companies generally don't say what they're doing. We have former PhD students from our lab working at some of these companies, including at Moderna, but they're not at liberty to tell us much. But I think that they are. But it's all kind of secret what exactly they're doing. Yeah.
So we do have a couple questions online, and I know we still have one in the room. So Masato Yamamoto asks, are the phase 3s for patients who have failed Pembro therapy or open to those that have not been treated with Pembro?
It's a question about enrollment on that trial, whether you can enroll if you've already received and failed on checkpoint. I don't know for sure. We can look it up. My suspicion is that they would not. My suspicion is that they're enrolling patients who have not yet received Pembro, and they're going to receive for the first time along with their vaccine. And some of these trials are doing a direct comparison. So there's one arm where they get the checkpoint and the other arm where they get the checkpoint plus vaccine to try to like really prove, like, does the vaccine add something here? Does it sort of synergize with the checkpoint therapy when you kind of give an instruction manual to the immune system along with the sort of unleashing of the immune system, taking off the brakes?
Can I just ask, I think one of the future efforts you were going to take was just looking at single cell RNA-seq data sets for mutation and neoantigen calling. Can you just comment on how mature that is and how promising you think that would be?
I don't know that we would use it for the mutation calling. We probably wouldn't replace the existing data set. It would be more additive to kind of, and we have done this already with like some test samples to, as a way to characterize the immune response. As it sort of like supports the ELISpot kind of readout or the manifest readout, where you're doing a similar experiment where you stimulate with either individual peptides or like the whole vaccine. And then you do single cell characterization. So you can start to match up like this peptide stimulates these T cells of this type, producing this CDR3 sequence, which I can see as an expanded clonotype. And then I can get the alpha and beta pairing because I have a single cell data. I can clone that TCR now. I can do like follow-up, like really like validate that these T cells recognize tumor cells, that kind of thing.
More on the reactivity side, yeah.
Yeah.
Okay, and then I have one question from Manish Patel. He says he's always felt that gene fusion proteins would be more immunogenic. At least in lung cancer, the recurrent gene fusion doesn't typically respond to immune checkpoint inhibitors. Any evidence that vaccine strategies might be more effective in these patients?
I don't have any evidence, but I agree that it seems like they should be really promising targets. And there are some people very focused on this part of the problem. So one example I think is really cool is the DNA JB1 PRKACA fusion, which has become essentially diagnostic of fibrolamellar HCC. Just basically in all of those patients, you get like a deletion that brings together these two genes and you get this fusion. And there is a clinical trial targeting that fusion specifically with a neoantigen therapy that's underway right now. And I'm very curious to see if that's successful.
I think we are over time, obviously. So one more round of applause for a great presentation. Thank you.